Hello and welcome to this video on the different levels of laboratories. These are known as biosecurity, physical containment, and biosafety levels. Although the exact term varies, the meaning is the same. They all apply to a laboratory, regardless of its use. This could be teaching, diagnostic, or research. The designations have been a point of public interest over the last few years. The most exciting, interesting, and dangerous research is conducted in the highest level laboratories, but the lowest level laboratories are to be found everywhere, which may take you by surprise. Nearly all laboratories that are involved in biological research of one kind or another are covered by these particular designations. This covers everything from microbiology with bacteria to viruses, some of the most dangerous historical pathogens such as Spanish flu and bubonic plague, cells for cancer research and propagating novel species of fungi, genetic research into GMOs, developmental disorders, acquired diseases, gene expression, and more. Even some of the more dangerous chemical research has to be conducted in these sorts of laboratories. As you may imagine, when genes are modified, there are dangerous organisms and possible genetic contamination, and caution must be taken. This is generally the focus of most biosecurity levels. At the same time, not all research is incredibly dangerous or risky. In fact, some applications of a laboratory aren't dangerous at all for the most part. Some of it is as simple as growing everyday bacteria to test novel antibiotics, find molecular genetic pathways, or even just identify what organism has caused an infection. In other cases, genetically modified organisms such as mice and crops must be contained in an appropriate laboratory. Depending on what is being done, the requisite level of security will vary. There are 4 levels of biosecurity. The 4 levels are fairly straightforward, 1 through 4. Each level gets sequentially more severe in the restrictions and controls, what's required to contain things like microbes and biological agents. Generally speaking, the primary determinant of what level is given to a laboratory and what containment procedures and similar must be taken is things like the severity of the disease being investigated, how transmissible it is, and the nature of work being done. In other cases, it may be the origin of it or the agent or pathogen in question. For example, you don't want Spanish flu being worked on in a level 1 biosecurity lab. This would allow far too easy escape and spread of what has been one of the most damaging diseases in our history. This is why you go from Biosecurity Level 1 through to Biosecurity Level 4. The most extreme restrictions are applied to Biosecurity Level 4. If you're in a lab that has the Level 1 designation, the microbes you're going to be working with are generally known to have no consistent cause of disease in a human, and so if you're a healthy, otherwise fit individual, there should be no risk to you whatsoever, and the chances of the disease breaking out and causing harm to others are equally minimalistic. Examples of this are things like E. coli. Something like this might be found in, say, a food safety laboratory, where quality control analysis is conducted to identify if any pathogens somehow have found their way into the foodstuffs. In other instances, it can be used in teaching facilities such as universities and colleges. In most instances, whatever you're dealing with isn't going to be particularly dangerous. There are a range of standard laboratory practices. Most of this is fairly straightforward. Things like don't lick the surfaces, don't drink in the laboratory, clean up after yourself, and similar. But nearly all the work can be done on an open lab bench as there's almost no risk of any kind of spread, so long as you're wearing appropriate PPE. Generally something like a lab coat, gloves, and eye protection. The minimum requirements. The facility itself must have the basics available as well, this being a sink for washing your hands, and a door which separates the laboratory from the rest of wherever it's located. Working within the laboratory generally has a few rules associated as well. You can only use a mechanical or electronic pipette. In some instances, other laboratories would allow the use of a mouth pipette. This is a rather obsolete practice, but in some instances applied. Things like sharps should be handled appropriately and disposed of appropriately. the work surfaces should be decontaminated daily, if not more frequently. As mentioned, there should also be the fairly standard rules, such as no drinking, smoking, eating, your fairly common sense practices. After all, who wants to have a mouthful of E. coli covered sandwich in the middle of a laboratory? Going up a level, you get to Biosecurity Level 2. This is also known as your Physical Containment or PC2 laboratory. This is the next level up and is used for microbes that may pose some hazard, particularly to people who, say, might be slightly weaker, older, or otherwise sensitive to infection, that there is some modest risk of danger to the environment should it escape. Generally speaking, these are your normally occurring pathogenic organisms. Think the more aggressive strains of E. coli that cause things like gastroenteritis. The reality is that these occur naturally within the environment, but at the same time, people are already exposed to them, and so they're only at risk under certain circumstances. Staph aureus is another good example of this in practice. Biosecurity Level 2 is also used in the case of genetically modified organisms for the most part. This is generally accepted as the minimum for a genetically modified organism. The reasons are simple. The access to the facility is extremely limited, especially when work is being conducted. That allows for not only containment of the facility, but also there's restricted access, which means people aren't likely to bring in or take out anything pathogenic or that shouldn't spread its particularly modified genome around. If you had, say, a mouse that escaped from a containment facility that was genetically modified, it might lead to it breeding, and that gene could be carried on to other organisms. The same basic level requirements will be carried over from your Biosafety Level 1 laboratory, things like PPE. But on top of that, you have added requirements. This includes the use of a biological safety cabinet. This is, in fact, very similar to a chemical fume hood. The differences are largely around the fact that it doesn't draw the material away as readily, often also contains things like UV lights that would destroy anything that's in there between uses. Further to that, material and equipment that is used should be put through an autoclave or other method of decontamination. Where Biosafety Level 1 only required that a door separate the laboratory from anywhere else, Biosafety Level 2 goes up a level again as well. And in this case, the doors must be self-closing. There must be a sink and eye wash facility readily available. This is important in the case of, say, a contaminant or pathogen getting into your eyes through a splash or aerosol. Notably on top of these requirements is that generally it should be a negative pressure environment. That is, air from the outside environment will be pulled into the laboratory area, rather than air from the laboratory leaching out into the outside world. This ensures that what goes into the laboratory stays in the laboratory, much like Vegas. Since you're working with more pathogenic organisms, this is where biohazard warning signs should start showing up if they haven't already. Generally speaking, biohazard warning signs would indicate that what you're dealing with is likely to be a concern. Going to the next level is Biosafety Level 3, Physical Containment Level 3, or Biosecurity Level 3. This has all of the requirements of Levels 1 and 2. This laboratory level is where things start to get interesting. And by interesting, we mean that it's pathogens or other things that could be considered dangerous despite being indigenous or part of the normally occurring flora and fauna of your body. The problem is they could potentially be lethal, and this is through something like respiratory transmission, coughing, sneezing, respiratory droplets landing on you and passing through. Because everybody has to breathe, this is the most obvious possible way a pathogen could find its way into you. Tuberculosis, or to give its proper name, Mycobacterium tuberculosis, is found in these sorts of laboratories. That's because it's passed via respiratory transmission, but importantly, it is extremely hard to treat and is extremely dangerous. On top of everything we've already mentioned, there are a number of requirements for containment within these facilities and use of them. Staff will often have to undergo regular medical checkups to ensure that they haven't been infected, and if it's possible, they'll be immunized for anything they're dealing with well before they start working there. As was the case with Level 2, access is extremely restricted and in fact often controlled based on when you should be accessing it and how. You're still required to have all of your standard PPE, and you may even need to be wearing a respirator the entire time you're working within the laboratory. You also still need to continue with the appropriate biosafety cabinets. What changes is that rather than just having a sink, you must have a hands-free sink and eye wash facility that are available near the exits of the laboratory. Much like a PC2 level facility, air is a consideration, but in this case, exhaust air cannot be recirculated back into the laboratory, and so air must have a continued directional airflow that draws fresh air into the laboratory from clean areas, and in doing so, it avoids bringing air in from potentially contaminated areas. This would prevent the spread of any diseases should they somehow get out of containment. There should be at least 2 doors that allow for an airlock type facility that further facilitates this directionality of air movement. Some further requirements, depending on where you're operating and the specific requirements of that particular legislative structure, may require that rather than wearing a lab coat, you instead have wraparound gowns, scrub suits, and/or coveralls when working within the laboratory. The final level is Biosecurity Level 4, and this is perhaps most often the image portrayed in Hollywood films. Blow-up jumpsuits, full helmets, and hooked up to an oxygen supply. All of this is to create an environment in which the researcher is completely isolated away from the things which are being worked with, and in doing so they are protected from what is being worked with or on. Level 4 continues to build upon all the requirements we've already mentioned, and being the highest level of safety, you would expect that they take it up several levels further. There aren't very many Level 4 laboratories across the globe. The reasons for this are relatively simple. They're incredibly expensive to build. There's not a lot of demand for what they do, and what they do is incredibly dangerous. These sorts of laboratories are for the most dangerous, exotic, and high risk of aerosol, blood, and other infective agents. Anyone who is infected by these has a very high chance of dying, even if they are treated for it. Diseases like Ebola fall into this category, and the reason they fall into this category is just how risky they can be if you're infected by them. One of the first and most obvious requirements if you find yourself in one of these facilities will be that you need to change your clothes before you can enter the facility. This ensures that what goes into the facility stays in the facility except for yourself. You and any materials you may have with you will also be required to undergo decontamination. For yourself, this may be as simple as showering thoroughly. When we're talking about the facility proper, all the work done within it is generally done within a Class 3 biosafety cabinet. This is a box in which you can reach in via gloves, and everything within there is completely separated from your particular environment. It means everybody outside that box is safe from what's within it. Alternatively, this is where those jumpsuits come in. It's a full-body suit with air supply going into it, and there must be positive pressure. This means the suit is constantly being pushed out and away from your body. If, for any reason, there is some sort of damage to the suit, air will be pushing whatever's trying to get into the suit away from it and out of that hole. Exactly like a balloon being punctured, or a ball or car tire with a small leak in it. Air constantly comes out and pushes things away from the suit, meaning nothing can enter it. The laboratory itself is even more difficult, and we've already mentioned that these are incredibly expensive facilities. One of the reasons for this is that they must be a separate building, or in an isolated section of an existing building. Access to this must be restricted to some of the highest levels possible. The laboratory itself must have a dedicated supply of air, as well as a dedicated exhaust system. This exhaust system must allow for decontamination of all the air that passes through it. Further to that, any physical matter going through that air system has to be vacuumed out. This is perhaps why when Hollywood depicts some of the most advanced laboratories working with extremely dangerous diseases, they choose to go to the PC4 facilities. They look impressive, but because of how difficult they are to enter, how difficult it is to use things within them, and often how hard they are to maintain, they're not a common occurrence. In fact, your PC2 and PC3 laboratories are some of the most common levels found for research purposes. The reason for this is straightforward. It's much easier to maintain these facilities. They can be built within existing infrastructure. PC2 facilities are often more than adequate for purposes that are needed by the vast majority of researchers. Those that need more can generally find a PC3 facility with relative ease compared to PC4. The Biosecurity Level 4 facilities, because they work with such dangerous material, are incredibly restricted, generally only ever state-run, funded, and owned. This is perhaps why places like Russia, when they had an outbreak of fire in one of their facilities, was frowned upon, why lax standards in other countries when it comes to following the rules around the running of these facilities are seen very negatively. It is simply this: the levels of biosecurity correspond to the threat to people, environment, and, as some may argue now, the world. Thank you for watching this video. If you have found it interesting, consider liking, sharing, and subscribing. Please post any comments, questions, or suggestions that you have below.